I told you this story because it nicely illustrates my first point, that we don't have to choose between action and inaction. And the reason we don't have to choose is because this choice has already been made for us generations ago, millennia ago. Think about where this story began. Why was there a meat crisis? Because we'd become dependent on cattle. This dependence began more than 12,000 years ago, when our ancestors started the process of transforming aurochs, the wild progenitor of cattle, into the tamed cattle that we know today. First, by changing our hunting practices, taking only males or non-reproductive females, leaving those that could continue to grow the population. Then by bringing these herds closer to our settlements, then choosing which individuals would breed and which would not. And we got better and better at this over time, until today, our planet is home to more than 1 billion cattle. That's 1 cow or bull for about every 8 people. To make room for all these cattle and for our other domesticated plants and animals, we took down forests and took over fields, transforming landscapes in the process. And as we moved into these newly transformed habitats, we often brought with us other species, either because we liked them or, as with rats and brown tree snakes, because they hitched a ride. Sometimes these species had no natural enemies to slow their growth, and so their populations exploded, often harming native species in the process. And of course, our plans to solve these crises often involved introducing other potentially invasive species without thinking too hard about the ecological impacts of doing so. But that's not all. We'd also begun to manipulate things by trying to protect them. Burnham wanted to bring in African animals to put them in national parks, to encourage people to visit the parks and therefore justify the existence of these new public spaces. Today, these protected spaces and the species that live in them are managed just as if they were another domesticated species. They're fenced in to protect from predators, they're vaccinated to protect them from disease, and their populations are counted and culled to prevent them from becoming too large populations or unmanageable. Conservation is not, and never has been, about leaving things alone. And then there was the solution. We ended up not needing hippos in the bayous because people just turned the land into better places for cattle. They reclaimed the land from the sea by dredging and let the grass grow, again, reshaping the landscape. We invented industrial scale agriculture and continued to improve our cattle, first through standard breeding to try to concentrate traits that we like in our cattle, and then through technologies like artificial insemination. This picture here is of frozen sperm straws collected from a prize bull. His frozen sperm can be used to create the next generation of cattle well beyond his own reproductive capacity or even years. These frozen sperm can be used decades after the animal who provided this sperm actually died. These technologies gave us increasing control over the evolution of our cattle. Today's cattle are highly refined animals that people designed to be particularly good at specific things, like this animal is to producing milk. Today, elite Holstein cows produce 25% more milk than they did 10 years ago, while requiring less food, water, and space. They are remarkable achievements of human ingenuity and design. But there are a lot of them, and there is a cost to us and to the planet of there being so many cattle. Each cow or bull can consume more than 18 kilograms, that's 40 pounds, of food every day. Methane gas released from their farts and burps, actually it's mostly burps, despite what you often hear, is responsible for nearly 1/7 of present-day global greenhouse gas emissions. And some of our cattle live in conditions that are decidedly unpleasant and even cruel. Which brings me to my second point. Today, we are once again feeling the strain of our successes as meddlers with nature. There are a lot of us, a lot of our domesticates, and we are, again, running out of space. People and animals are suffering. There is an undeniable climate emergency and extinction crisis. But once again, we have new technologies at our disposal. Our choice is whether to use them. I personally think that we need to use them. And to illustrate my point, I'm going to give you an example and stick again with cattle. If you look at this image, there's something that you might notice about these cattle. What is it? I would ask for guesses, but I can't hear you, so I'm not going to. It's going to be obvious once I point to it anyway. Many of these cattle, look at this guy right here in the middle. Many of these cattle, which are packed pretty tightly together in this picture, do not have horns. Some cattle, like these Texas Longhorns here, grow massive horns, and others, like this Angus cattle here, are naturally hornless. This is a trait also known as polled. Angus are an elite breed designed for beef, but lots of our important breeds are not naturally polled, and in many countries, farmers are mandated to remove their horns when they're young. In the US, around 15 million calves are surgically dehorned every year, and dehorning is unpleasant, expensive, and painful, and unsurprisingly raises significant concerns related to farm animal welfare. But there is another solution, and one that takes advantage of this new family of tools to which I have just alluded: genetic engineering. Alison Van Eenenum, a professor at UC Davis, has been collaborating with a biotech company called Recombinetics to use genetic engineering to transfer the gene that causes Angus cattle not to grow horns, the polled gene, to Holsteins, which do grow horns. Actually, it's not a gene, it's just an allele. There's different versions of this gene. Like, the different versions are called alleles, and Angus have a version, an allele, that causes them not to grow horns, while most Holsteins do have a version that causes them to grow horns. They have to be surgically dehorned. So this could be done the regular way. She could take an Angus bull and breed it with a Holstein cow, and their offspring would be hornless because they would inherit one copy of that allele from their Angus dad, and that would cause them not to grow the horn. But this would not be good because it would not be either an elite beef cow or an elite milk cow, because a full half of the DNA would be transferred. They would get a full half of their DNA from each parent. So they would be a half beef cow and a half dairy cow, and it would reverse all of these engineering that we've been doing over years and years as people. But with the tools of genetic engineering, Alison and the team at Recombinetics transferred just this one allele that evolved more than 1,000 years ago in the ancestors of Angus cattle to a Holstein bull. They then inseminated later a Holstein cow, and they transferred it, and then they cloned this bull. So they grew up this bull. His name was Bury, and he was a Holstein that was genetically engineered to have this allele from Angus. And then they collected sperm from Bury, and they inseminated Holsteins with his sperm, and they had six births, 6 happy, healthy births some months later. One of them is shown here, Princess, and she has, as you can see here, not grown any horns because she has inherited the polled allele from Bury, her genetically engineered dad. And here she is sitting next to her cousin, Cinderella, who you can see has grown horns. So clearly this genetic engineering has worked. No need to chop off any horns because of genetic engineering. It's amazing, I think. So how might we harness this technology to address other challenges that we face? Imagine if we could figure out how to engineer cattle not to release so much methane into the atmosphere when they farted and birded. Farted and burped, I can't talk. Well, we don't quite know how to do that yet, but there are other environmental problems that caused by our domesticates that we can solve now using the tools of genetic engineering. So this is a normal pig. Pig farming is possibly the largest agricultural industry in the world, and it is environmentally problematic. Pig growth is limited by the amount of phosphorus that they can ingest. So when a farmer feeds her pigs, she often adds phosphorus to its feed. The pig is inefficient at converting this phosphorus into something that's usable, and so much of it is excreted into their waste, where it finds its way into the local environment, causing eutrophication of the landscape. The enviro pig, however, solves this problem. The enviro pig is a pig, and its DNA is mostly pig DNA, but its genome also includes a gene from a microbe and a gene from a mouse. These 2 extra genes express a protein in pig saliva that breaks the phosphorus down into usable form. Enviro pigs can be fed less phosphorus, saving the farmers money, and they can use that phosphorus more efficiently, saving the watershed. The enviro pig, which was developed by scientists at Guelph University in Canada, was submitted to regulatory approval in 2010, but no one really knew what to do with this genetically engineered pig. The approval process stalled. There's not enough money in a lot of these public institutions to be able to push approval processes through these expensive and time-consuming mandates. And the project eventually ran out of money. Enviro pigs solve a problem that plagues one of the largest agricultural industries in the world. But our discomfort with the technologies that created them is thwarting the breakthroughs that this technology could bring. If we could find our way beyond this impasse, imagine what else we could achieve. Could we use the technology to save corals and black-footed ferrets, to rebuild forests and save island birds? Well, let me tell you a story about Elizabeth Ann. She was born last December, and she's, let's face it, pretty cute, right? But she is also much more than that. Elizabeth Ann is a black-footed ferret. The favorite prey of black-footed ferrets is prairie dogs. Prairie dogs are grassland herbivores that are notable for their capacity to destroy crop and farmland by digging elaborate and connected burrows. And throughout the 20th century, were subject to pretty successful eradication programs that not only eradicated prairie dogs, but also, accidentally, their predator, the black-footed ferret. In fact, black-footed ferrets were among the first listed species when the Endangered Species Act first went into effect. And in fact, they were thought to be entirely extinct after a captive breeding program failed to make more black-footed ferrets from a population that was found in the 60s and 70s. And then in the mid-80s, a population was found near Meeteetse, Wyoming, and this gave folks in the US Fish and Wildlife Service a second chance to figure out how to breed them. And they did. This collaboration, new technology, allowed them to use this captive breeding population to make way more black-footed ferrets. And it was good. It was great that they were finally able to do this because in the wild, black-footed ferrets were facing two very distinct challenges. First, there was only one population left. And by the time that this population was discovered, there were very few individuals that were still alive. And many of the individuals that they were finding were getting sick. And that's because introduced sylvatic plague had been infecting the prairie dog population. So whenever a black-footed ferret found and caught and ate a prairie dog, it got sylvatic plague and it died. How can we use our new technologies to save black-footed ferrets? Turns out there's also two possible solutions. And this is a program that's being coordinated by Revive and Restore, which is a biotechnology-based nonprofit focusing on conservation. It is based outside of San Francisco in Sausalito. Revive and Restore have been collaborating with lots of different institutions for several different plans. And the first solution that they came up with was to actually introduce diversity from the past. So there's this amazing resource in San Diego, California called the Frozen Zoo. It's part of the San Diego Zoo and led by a guy called Ollie Ryder, who's the person here on the right. And for at least 3 or 4 decades, they have been collecting tissue specimens from endangered species from around the world and putting them into deep freeze, where you can keep the tissues, the cells in these tissues alive for some time. And in collaboration with US Fish and Wildlife, the Frozen Zoo, and Revive and Restore, that is where we eventually ended up with Elizabeth Ann. Elizabeth Ann is a clone of a black-footed ferret that was alive more than 30 years ago. And more importantly, she is actually from a population that was different than the one that is currently being used in the captive breeding program. So when she is introduced into the captive breeding population, when she becomes reproductively mature, which she will be the 8th founder lineage. Currently, there are only 7, and the 7 are extremely closely related. So having Elizabeth Ann and having her DNA to introduce into this population is going to be amazing in terms of reducing the potentially devastating effects of inbreeding in today's captive bred and wild black-footed ferret population. It may not, however, be enough to save them from plague. And this is where we get to the second solution. The domestic ferret, which is a cousin of the black-footed ferret, is naturally immune to plague. So Revive and Restore, in collaboration with the Ehrlich Institute, the US Fish and Wildlife, the Smithsonian Institute, and many academic partners, are working to try to figure out what it is about the domestic ferret genome that makes them naturally immune to plague. And once they've discovered this, their plan is to use gene editing technology to transfer that evolved resistance to plague from domestic ferrets into black-footed ferrets in the captive breeding population, so that they can release black-footed ferrets into the wild that themselves are, because of the addition of this tiny bit of domestic ferret DNA, immune to plague. What is the chance that we are actually going to use genetic engineering to save a species from extinction? There are no currently existing genetically engineered black-footed ferrets. We still don't know what DNA it is that causes this genetic resistance. And so we're still at the very early stages of figuring this out. But this brings me to my final example here, the American chestnut tree. This glorious tree right here from this picture that was taken in the early 20th century. American chestnut trees were once the dominant keystone tree in eastern forests distributed across eastern North America and into Canada, the range here in green, but also extending a little bit further northward. In the early part of the 20th century, however, a fungal disease, fungal blight, was introduced into the American chestnut tree population in shipments of trees from Asia. And this fungal disease releases acidity in order to burn holes through the black-footed ferret, through the American chestnut tree, through the tree's trunks. And this acidity causes the fungus can actually make its way through these holes that it's burned through using the acidity, but it causes these cankers to start to form on these trees. And eventually the cankers throttle the tree like a big belt, and the leaves start to fall off and the branches fall off, and eventually the whole tree dies. And over the course of just a few decades, 1 billion American chestnut trees succumbed to this fungal blight. But they didn't die entirely. The roots actually can survive underground, and still today some of these roots will shoot up what I like to call zombie trees that grow for a little bit, rarely enough to actually produce any acorns, but they do grow a little bit. They don't look that great until eventually they too succumb to the fungal tree and they die, fungal blight, and they die. There are also a couple of populations of, I call them expat trees, that were planted by people as they moved across North America to establish colonies throughout the 18th and 19th centuries. And these trees are starting to become sick with blight as the fungus is making its way across the country, but they also produce chestnuts. So what's cool about this is it allowed Bill Powell, shown here, and Chuck Maynard to begin a program to create genetically engineered American chestnut trees using the living material that they have from these shoots that come up and from these expat populations of trees. They looked to try to figure out what they could do to neutralize this fungus. And it turns out that lots of plants have developed an approach to neutralize fungi that infect them over this time. So they picked a gene from wheat. Lots of plants make it, but they picked wheat because we eat a lot of wheat, and therefore there shouldn't be any worries about eating this wheat gene if we decide we want to eat a genetically engineered American chestnut. And they engineered an American chestnut genome to contain this gene from wheat. And what it does is it excretes an enzyme, it codes for an enzyme that neutralizes the acidity that's produced by the fungus. It doesn't kill the fungus, but it stops the fungus from killing the tree. And this genetically engineered tree that they call the darling breed, the darling lineage, this strain or darling strain of tree, is now going through the process of regulatory approval in the US. It turns out too that you only need one copy of this gene, so they can take this engineered tree and outbreed it with all of the surviving lineage of trees that are there, really bringing back all of the diversity of the American chestnut tree that used to exist, but with this one slight tweak, this addition of a tiny, tiny bit of DNA from wheat that allows that tree to survive and thrive in today's environment. So as part of this regulatory approval process, the tree is actually being regulated by 3 agencies in the US and also several in Canada. So this is not an easy path for them to pave. Part of this included last year a public comment period. They received actually tens of thousands of comments, and most of them, we were all happy to see, were positive. And here is an example that I love. This is a comment from James Sneed, who is a tribal council member of the Cherokee Nation. He writes, and I'll just read out loud the bit that's in yellow here. "We, as an indigenous people, have relied on the chestnut tree to provide life to our people for most of our existence. When the blight was introduced to our land, our way of life was changed, and not in a good way. I hope that the new tree will show that we, the human race, are trying to become healers of our land and help restore such a vital resource to a people that have relied on its gifts of life and shelter for so many years throughout time. Healers of our land. The point made by James Sneed that I agree with and that I hope we can all see or begin to see is becoming important. We need to get our hands dirtier. We need to learn how to meddle better. We are a long way from having the American chestnut trees as the keystone species that once dominated the eastern forests of North America. But with the genetically engineered darling tree, we're on our way.